Students, now discuss about soluble mediators of immunity. These are the mediator act as a mediator in the immunity, body immunity. These are soluble mediators of immunity. Students, these are three types. Soluble mediators are three types. One is the complement proteins, and second one is cytokines, and third one is antibodies. Students, first about the complement proteins. What about the complement proteins? Complement proteins are plasma proteins present in the form of the plasma proteins, also cell surface proteins, okay, and activates. These are the complement proteins which are available in the form of plasma proteins, cell surface proteins or activates, and forms the membrane attacking complex, MAC. MAC means membrane attacking complex, students. And this is the membrane attacking complex attacks the cell membrane of the infected cells, okay, and forms the pores or the apertures on the cell membrane, which the extracellular fluid is allows into it through the pores. ECF entered into the infected cells or the target cells, and finally this leads to swelling and the burst of the infected cells. Next, student, another some amount of the complement proteins also forms the coat on the surface of the pathogens. Where the pathogens are available in the body, these are the complement proteins also forms the coat-like arrangement on the cell membrane, on the membrane, on outer surface of the pathogens. After coat formation by the complement proteins, attract neutrophils and the macrophages. After the formation of the coat on the microbes or the pathogens, these are the complement proteins attracts neutrophils, macrophages to phagocytosis. Okay, student, or cytolysis. Lysis of the cell. Lysis of the infected cell is called cytolysis. Okay. Neutrophils shows effective action on the microbes by phagocytosis or cytolysis. This entire process is called opsonization. Okay, student. Okay, opsonization is the one of the process in the immune system to destroy the infected cells by the neutrophils, macrophages. Next, students, second one is cytokines. What about the cytokines? Cytokines secretes by T helper cells. These are the simple soluble proteins present in the body fluids. Cytokines secretes by T helper cells and bind to cell surface receptors. These are the cytokines bind to the cell surface receptors. Okay, cell surface consists receptors. These are the cytokines bind to the cell surface receptors and initiate cell surface receptor. These are the receptors present on the lymphocells. Okay, these are the cytokines bind to the cell surface receptors of the lymphocytes and initiate differentiation of the cells of immunity. Which cells participates in the immune system? Which cells plays main role in destruction of the infected cell, destruction of the antigens, removal of the antigens in the body. That type of the cells goes to the differentiation, okay, by the cytokines, okay, to stimulate phagocytosis and cytolysis of the infected cells. Okay, student, and these are the cytokines also stimulate phagocytosis and cytolysis of the infected cells. Okay, student, and these are the cytokines are available in two types. One is the interleukin, second one is interferons. Okay, interleukins, these are produced by leukocytes, white blood cells, and causes a differentiation of the immune cells. Immune cells goes to the differentiation by the cytokines. Which are cytokines? Interleukins are the cytokines that is helps to differentiation of the immune cells. For example, TC cells differentiated into cytotoxic T lymphocytes and effector T cells. This is possible by the interleukins. And interferons, what about the interferons? These are the antiviral proteins produced by the virus infected cells to protect the neighboring cells from the virus infection. Okay, student. Next, another, it is the most important, student, that is antibodies. Antibodies also called Ig, immunoglobulins, represent with Ig, immunoglobulins. Okay, these are the antibodies inactivates antigen. Okay, student, antibodies, these are produced by B lymphocytes. B lymphocytes produce antibodies when the microbes enters into the body. Okay, these are identified by the immune system. At the same time, B lymphocytes produce army-like proteins to fight against the antigen particles. Okay, student, the type of the proteins are antibodies produced by B cells. These are proteins. Okay, and these antibodies are highly specialized for binding with specific antigens. Okay, these are the antibodies are fight against specific antigen only. Okay, specific antigens binds with the specific antibodies. It is important, student. Okay, one of the antibody not shows the fight against the all type of the antigens. Okay, one of the antibody that is produced by the B cells shows fight against the specific antigen only. Next, paratope. What about the paratope, student? It is important. Student, listen carefully. Paratope, antigen binding site. Antigen binding site. Okay, antigen binding site. It is the part of antibody. Paratope is the part of antibody. Okay, that is the site which binds the antigen particle and recognize antigen. This is the paratope of antibody, also recognize the antigen. Student, it is the paratope. It is the paratope. This is the site of the antibody that recognize the antigen and binds the antigen. So it is called antigen binding site. And based on the mobility, these are the antibodies are two types. One is the circulating antibodies and second one is surface antibodies. Okay, so circulating antibodies freely circulates in the body fluids, in the lymph, in the blood. Okay, student. Next, surface antibodies. These are the surface antibodies are attached to the cell membrane of the B lymphocytes and the memory cells. Okay. Next, and structure. What about the structure of the antibody? Immunoglobulin given by Rodney Porter. This is the structure is represented by Rodney Porter, scientist. Okay, and it is the Y-shaped molecule. It is the Y-shaped molecule, student. Y-shaped molecule. 4 polypeptide chains present in structure of the antibody. 4 polypeptide chains, student. In these, 2 are long heavy chain, LHC, long heavy chain. Student, it is the one of the long heavy chain. It is the another one of the long heavy chain. Next, 2 short light chains. Student, it is the short light chain and it is the another one. Okay. Next, so it is represented with H2L2. H2L2. Next, 2 chains in this antibody structure. 2 chains are linked with. Okay, it is the AV chain. It is the AV chain. It is the light chains. Okay, these 2 are linked with the disulfide bond. It is the disulfide bond. It is the another one of the disulfide bond. Okay, student. V means variable region and C means constant region. Okay, and based on the structure, these are the immunoglobulins. These are the antibodies are 5 types. Okay, student. Immunoglobulin D, immunoglobulin E, immunoglobulin G. Okay, and immunoglobulin A and immunoglobulin M. Okay, student, based on the structure. Okay, based on the structure, these are the immunoglobulins are divided into 5 types. Okay, it is the Y-shaped structure of antibody, AV chain and light chain. Okay, both are combined with the disulfide bond. Okay, student. Next, it is the paratope. 2 sites that shows by the each of antibody. One is the antigen binding site, Fab, student, that is called Fab. Okay, Fab means an fragment of antigen binding site. Okay, it is the antigen binding site of the antibody. So it is region is called fragment of antigen binding site. Okay, it is the FC end, student. FC end that is attached to the cell surface, cell membrane of the B lymphocyte or the memory cell. So it is called fragment of cell surface. It is the region, fragment of cell surface end. Student, it is about the soluble mediators of the immunity. One is the complement proteins, another one is cytokines, and third one is antibody.